Hello, I'm going to be going through Tuesday 12th of January 2010, the Unit 1 paper for Edexcel Biology. I'd recommend that you have your paper with you, the reason being that rather than having the camera just showing the paper, or rather than having to read through each of the individual questions every time I go through them, it'd be much easier if I just sort of talk to you and you can have a look at the paper with you. Also, what might be a good idea, and what I always have, is I always have the mark scheme for my particular paper open at the same time. If you're looking at the mark scheme straight away, your answer is still fresh in your mind. You might have had something that you couldn't quite remember, and looking at the mark scheme immediately afterwards will help you to remember, that's what was just missing in my head, that's what was so close to me getting the full marks. And hopefully, because it was so close and because it was so fresh in your mind, hopefully it will help you to remember it a bit better the next time. This is here to help you with all of your papers, so hopefully this shouldn't just help you with the Unit 1 of January 2010. This is not your mark scheme. Hopefully this shouldn't even just help you with Unit 1. Hopefully it should be able to help you with all of the different papers that you could potentially come up against doing, because I'm going to be talking about various different strategies to use throughout your paper, about particular questions and about revision in general. So have your paper with you and we'll have a go through it. Right, the first question is a multiple choice section about your DNA replication. This is quite common in the Edexcel papers to have to start off with some multiple choice questions. Sometimes that can be a bit of a relaxing thing for people to get into. Right, structured label J is deoxyribose. It's not a ribose. That's a really common mistake that people make. This is a pure knowledge section, but there are questions in here that are going to trip you up just because you're going to be like, oh yes, it's a ribose, I know this, quickly move on. Read through all of the options and you'll suddenly realise when you get to the bottom, that was close, I very nearly ticked ribose. Okay, structured label K is a phosphate group. It's not a phosphorus atom, that's the only thing I could potentially think that you might get wrong. The phosphate group actually contains phosphorus and some oxygen in there as well, which is why you can't just call it a phosphorus atom. The bond labelled L is a phosphodiester bond. Structured label M is a mononucleotide. Now E, if the base labelled N on the parent DNA molecule is adenine, the base labelled O on the new DNA molecule will have to be thymine. It's DNA. They've given the option there of uracil, which is the replacement for thymine on an RNA molecule. If this question was about transcription and translation, then that would have been your answer, then uracil would be the answer. The bond labelled P is a hydrogen bond. It's always represented by a dotted line. Okay, now moving on to question 2. What's a quite good strategy to use with your questions is, as you're reading through them, you could potentially start to underline or highlight or circle different parts of it that would help you to focus your mind on different sections. Because one of the biggest things that people lose marks on in their past papers is not using enough keywords. They know the answer, but they give overly simplistic answers. It's very common that people will be asked a question about how is energy generated in the body, and they will forget to use the word respiration. So hopefully underlining and circling things will narrow your search in your memory down to some certain areas, and hopefully those keywords will then be triggered before you've even started looking at the question. This is the only opportunity you're going to get to show off to your examiner. Use it. Put down as many keywords as you can. This question in particular isn't particularly helpful, it's just a fill in the gaps exercise, but you do have to use the most specific keywords you possibly can. Okay, so read through the following passage on protein synthesis. I would personally circle the word protein synthesis. Right, so protein synthesis involves 2 stages. The first stage is transcription. A lot of people get confused between transcription and translation, and they forget which one comes first. If you sort of dissect the word, I find it quite helpful to dissect words quite a lot of the time to help you remember what they mean. Transcription is about scribing, it's about rewriting something. If you think of DNA and mRNA as being two different forms of the same language. What you're basically doing is you have your original information on the DNA, and you can imagine it as a very precious book that has to be kept in a library. You're not allowed to take that book out of the library, otherwise it will get damaged. So you go in and you rewrite the information by hand, or you photocopy it, and what you're basically doing is transcription. And that's what the body's doing. The DNA is too precious to be taken out of the nucleus because it would get damaged. Therefore, the body copies it using the form of mRNA. So mRNA is basically your handwriting or your photocopy. It is then taken out and translated, and translation is language, changing the language. So what they're now doing is they're taking the language of mRNA. So mRNA and DNA, they use the same language, mononucleotides. They then take that outside of the cell and they translate it into the different language of amino acids and into the protein. And takes place in the nucleus of a cell. During this stage, a molecule called mRNA is used. mRNA is made using the antisense DNA strands as a template. The second stage, known as translation, going back to what we were talking about, takes place in the cytoplasm of the cell on structures called ribosomes. During this stage, tRNA molecules, again, it's not good enough to just say RNA, tRNA molecules enable the amino acids attached to them to line up in the correct order. The amino acids are joined together by the formation of peptide bonds. It's really important to know about your biological molecules. So, peptide bonds, it's glycosidic bonds for carbohydrates, it's ester bonds for fats. Make sure that you are aware of how they form, of the kind of relationships they make, of the kind of bonds they make. There should be information on Canvas about it. There should be a poster on there somewhere, I'm sure. If there isn't, email me and ask me for it, because I've got it, I wrote it. So. Right, question 3 is the first of the graph questions we've got in this particular paper. Graph questions can be quite difficult to get marks on if you aren't used to them, and that's why it's very, very important to do loads of exam technique practice, because you can read through the textbook as much as you like, they won't have the examples of the kind of graphs you're going to get in the exam. And that's a good thing, because we're trying to train people to be scientists here. We're not trying to train people to be able to read a textbook. Lots of people can read a textbook. The important thing is being able to get new data and understand it and analyze it, basically. So again, having a look through your question, if you start to highlight various words, so for example, substances in the water can enter the cell by a variety of mechanisms, hopefully that will trigger off ideas in your mind. Ooh, I reckon this question is going to be about diffusion, active transport, maybe osmosis, that kind of thing. Really look at the graph and make sure that you understand it. Read through the graph, understand what's actually happening. It's very, very important before you've even read the questions to start thinking about that, because the questions can sometimes make you develop preconceived ideas. So you might look at the question and then go back to your graph, and rather than looking properly at the graph, you might assume that it's going to say something already. If you have already read the graph itself, hopefully you will have less of a preconceived idea about what's actually happening in the questions. Look at it very, very simply. They are the same. Substance A and substance B have the same rate of uptake for the first 30 minutes. Okay, the lines are so close together, they are basically connected. Then B, the uptake of substance B slows, and by the time you get to around 2 hours, B is actually leveled off. A continues for the same rate throughout the experiment. One good insurance point that you can tend to give for most describe the graph questions is called manipulation of figures. So, for example, if you go to your graph, you could say that at 2 hours the difference between the uptakes is 1 microgram per centimetre cubed, whereas after 4 hours in total, a further 2 hours, it's the difference between them is 6.75 micrograms per centimetre cubed. So you are comparing them in a real way. You are actually using numbers to show something useful. Question B is actually the first time that you get the quality of written communication marks. You can actually write your entire A level in bullet points. It's perfectly fine. If you start running out of time at the end, absolutely bullet point everything, because it will save you time. So for this one, check the number of marks. You've been asked to describe and explain how the results of this experiment support the statement. You've got 4 marks available. Always check the number of marks, because it will inform how many separate points you need to make about the particular question. The first thing to do, I would recommend, is to actually define the word diffusion, because if they're asking specifically how the graph supports the idea of diffusion happening, how else are you going to explain? So basically, diffusion is net movement of particles down a concentration gradient, and you could say that this supports this statement, the graph supports this statement, because substance B begins to slow down as the gradient begins to decrease. So if you've got a gradient like that, diffusion is going to happen very, very, very quickly down it. It's like a car going down a steep gradient. As the gradient starts to level off, as you're getting more of the substance on this side and less of the substance on that side, then the diffusion is going to happen slower, and that's why the graph is leveling off. Then it actually levels off completely, so diffusion actually stops, or net movement of particles actually stops, when the concentration has equalized. Right, substance A then, so talking about active transport and diffusion, this is a very, very common question. I would recommend you learn keywords and definitions for these kind of things. Those definitions actually have all the keywords in them that you could ever possibly need, and you will probably be using those definitions as you're answering questions about the 3 types of movement throughout cells. So once you've understood those definitions, once you know those definitions, you'll easily be able to list 2 or 3 differences between active transport and diffusion. Another thing just to point out, for some of you this may be really obvious, but it's 2 differences. You can't say one difference is that active transport requires ATP and the second difference is that diffusion is passive. Those are the same marking point. Right, the next question here, question 4, is about the different ways that organisms can carry blood around the body and can supply the body with oxygen and glucose and various other essential nutrients. Now, notice that I've missed out 4A because that is literally just a memory questions, and you have got the mark scheme. Now, this is a classic example of the way that the Edexcel syllabus expects us to understand completely about the human circulatory system, but it then in the exams asks us to do work about fish circulatory system and insect circulatory systems and potentially many other different kinds of circulatory systems. Now, this may seem to some completely unfair, and it does put people off when they start taking the exams, because it makes them panic and think that, oh my god, I didn't revise this, how can I possibly know this? They expect scientists to be able to take something that they already know about one system and apply that knowledge to a completely different system because of the parallels already inherent in the 2 systems. So it isn't unfair, and it isn't something that they're trying to catch you out on. They are actually trying to get you to use your knowledge in a different way, maybe a slightly unexpected way. So if you know, for example, that the human circulatory system is a double circulatory system, then you could probably predict that the fish circulatory system is a single circulatory system, and you can describe it accordingly. So you can describe how the heart pumps the blood into the gills and then straight around the body. Then you can now think about the advantages that the human circulatory system has compared with that of the fish. And that then brings you straight back to what you did learn in your syllabus, the advantages of the human circulatory system. You're just now putting it from the angle of comparing it with fish. So is that the heart is in 2 sections. You've got the right side of the heart, which is weaker, pumps just to the lungs, and the important part about that is that it's sending it through some very, very fine capillaries, and if it was under high pressure it would potentially damage those fine capillaries. So by having a double circulatory system, we are reducing the potential for damage to our lungs, whereas the fish is having to have a good pump from the heart to get it through the gills first and then around the body. Following from the lungs, the blood returns to the left side of our heart, which is so much stronger, and that pumps it much faster around the rest of the body because it doesn't have to worry anymore. Now, moving on to C, again, this is another situation where you've not studied in great depth the circulatory system of an insect. You can see that blood is just flowing into a cavity here and then passing back into the heart. So it doesn't actually have a system of blood vessels. Insects are very much smaller than us. And that means that therefore they don't have such a great diffusion distance. Now, if you can get words like that into your exam paper, then that sort of cuts out the need to say a lot more. Nutrients can diffuse very quickly into all of the cells without being actually taken to them by a network of capillaries and veins. Now, question 5 there is now a QWC question. Questions labelled with an asterisk are ones where the quality of your written communication will be assessed. You should take particular care with your spelling, punctuation and grammar, as well as the clarity of expression on these questions. Now, you would have thought then, from that explanation, from that sort of proviso on the front there, that if you don't write beautiful prose, that you might therefore get penalised. Now, that's not entirely true. On the mark scheme it says quality of written communication, spelling of technical terms shown in italics must be correct, and the answer must be arranged in a logical sequence. But what you could potentially do to help you with the spelling and also the remembering of these key scientific words is to write them on post-it notes, for example, small pieces of paper, stick them all over your house, and the more you see them, the more it will be taken into your mind sort of almost passively. The other thing is you need to get it in the right order. When you have a process to remember, such as blood clotting, atheroma, cardiac cycle, and that's just the ones in this topic. You've then also got the ones in Unit 2, Unit 4 and Unit 5. Don't forget then the core practicals. If you've got any of those things that could be really classed as a process, that's something where one thing follows another, I would really recommend studying them and learning them in a very chronological type way. Maybe it would be helpful if you drew yourself a flowchart of these things happening, simplify everything, draw them as a diagram where one thing follows another. There are quite a few of these processes where I've actually made flowcharts and made posters and diagrams for students. Some of them are already on Canvas, but if you come across any that you can't find on Canvas, that you're having trouble with, let me know, because I'm always looking for more to make. If you can get all of those concepts in there, that's great, but you don't need to. You only need to get 4 of those particular things to get the full marks. Obviously, when you're doing your exam, you don't know which things are going to be given the correct mark for. So don't limit yourself to just writing 4 ideas in a process question. In a process question where you have to write in chronological order, you might as well write everything that you know, just in case in the mark scheme they've put 2 concepts together, something that maybe you would have thought is separate, they might have put together and given you 1 mark for it. What is important though is making sure that you don't write too much. So for example, one thing that's very common is that students writing about the cardiac cycle. If they are given a question saying describe how the sinoatrial and atrioventricular node work in the cardiac cycle, they may go crazy explaining all about valves, all about backflow of blood, all about the double circulatory system. Basically, they are writing everything that they know about hearts. They won't get a single mark for any of those concepts. You need to limit your answers to what the question asked for. So, using the information in the graph, describe the effect of snake venom on the clotting time of human blood. Now, this is a massive thing. Again, I would seriously circle that word there, describe. So many people go on to explain what they think is happening and why the graph is that particular shape, and you won't get any marks for it. A lot of people spend so much time explaining the graph that they actually don't describe the shape of it at all, and that means that they end up losing, not gaining any mark. The first thing to do when you're asked to describe a graph is think simplest first. So what is the most obvious thing about this graph? Snake venom decreases the clotting time. All you're doing is sort of showing which direction the graph is going. Then start to talk about it in more detail. Only a very, very small increase in snake venom to start with causes a big decrease at the beginning. And after that, the line becomes shallower. So you're making the point that the important bit is at the start. So you can then use what's called manipulation of figures. So you're looking at the graphs, you're actually using the numbers on the graphs to illustrate your points. So you're talking about the biggest decrease that happens here. So in the first 0.004 micrograms being added of the snake venom, the clotting time of blood drops 60 seconds. Choose the most interesting differences, choose the most interesting change. Describe the change using figures, so do a subtraction. After you have done your description, do the manipulation of figures. There is usually only 1 mark available for manipulation of figures. It's always worth putting in there, but after you've done your verbal explanation. Right, suggest why the clotting time of the human blood with snake venom added was compared with the clotting time in the presence of factor XA. So again, you might not know anything about snake venom, but you might be able to figure out, well, they've got snake venom and they've got a normal human blood clotting factor. Maybe they want to use snake venom as a type of medicine. Maybe they want to figure out what type of medicine may be able to counter the snake venom. So they're talking about antivenoms here. Right, the component of the snake venom that affects blood clotting is an enzyme. Describe the structure of an enzyme. Keywords here. The most important keywords when you're talking about enzymes tend to be things like active site, tertiary structure, enzyme substrate complex. If you can try and get those into your answers, then you'll be sort of well on your way to getting your points. For this reason, it is a good idea to do loads of past papers online and to look at the mark scheme, because they have the major keywords and the same keywords come up again and again and again. So reference to enzyme as a protein. You think that's obvious? Stick it in there anyway, because it's only obvious because you know it so well. It doesn't mean that it's not correct. Sometimes it's really important to state the obvious. You don't know it because it's easy, you know it because you've worked to it. So enzyme as a protein, reference to the tertiary structure or the globular structure. Named bonds holding the structure in place, so like the hydrogen bonds, the disulfide bridges, and the ionic bonds holding the amino acids in place. So the bonds are between the R group. That's important because the hydrogen bonds, there are hydrogen bonds in the secondary structure as well, and the secondary structure is not at all important in enzymes. Well, not really anyway. Reference to the active site, idea of the specificity of the active site. Use as many keywords as you possibly can, because you don't know which ones they've got on their mark scheme. They might have had primary structure in their mark scheme. They didn't this time, but next time they might. Right, so the last question of this question 5 here then is suggest how the enzyme in the snake venom could be involved in the blood clotting process. Again, you don't know specifics, it's a suggest question. However, you do know how enzymes work and how specificity of enzymes work. Therefore, you could potentially be thinking, well, maybe actually the snake venom enzyme could be very similar to the enzyme that affects blood clotting in the human body. So it wouldn't be a great stretch to then talk about it having maybe a similar active site, maybe have a similar structure, similar tertiary structure, and then potentially it could activate other enzymes in the clotting cascade. Right, now moving on to question 6 about caffeine and about different caffeine levels in different drinks. It's very, very basic ones that maybe each of the tea or coffee samples could have different caffeine contents. You're just literally saying coffee has the highest amount, white tea has the lowest amount, or again, you could potentially manipulate figures to show the, let's say, the difference between coffee and white tea. The important bit that I really wanted to talk about was B, which is talking about an experiment using Daphnia. This is so important. It's the absolute reason why you really need to know your core practicals. Okay, they're all on Canvas. There is a resource very near the top of the modules section about the equipment that you need for the core practicals. So potentially you could actually undertake these core practicals yourself. If you are finding it hard to get the equipment, then you just really need to go onto the suggested web links that I've given you on that same document and find out how the practicals are actually undertaken. You could find some of these practicals done on YouTube as well. YouTube is a very difficult thing to put as a resource on Canvas because they do tend to take videos down and put better ones up later. Just do a search for Daphnia caffeine experiment. You will probably find some good videos on there. You'd be surprised at how many people upload their own videos of their science experiments. So know your core practical. Learn this as a process, a step-by-step method, because it's really common to get a question like this. It may have a different hypothesis on the practicals that you're reading in revision. Be prepared for them to give you or to ask of you a variation of what you've already learned. Make it more detailed in things like how you're actually going to determine the heart rate. But the idea, the marks that they've given on the mark scheme, the idea that heart rate must be determined before treatment. So you've got to get a resting heart rate with something. That's almost like your control. Then the Daphnia need to be put into the tea and allowed to acclimatise. There's no point adding the tea and going, oh, quickly, it will instantly have. Notice here that you will get 1 mark for writing out some equipment. So it is worth listing the most important equipment, and it is worth listing the most important things like the variables, how you're going to actually get the results, and then very important things like repeating results and so on. Next bit is ethics. So the friend didn't agree with using Daphnia in the experiment. Ethics can be another thing that students hate getting questions on. I think it's quite a good idea to get these kind of answers from looking at exam papers. So this is again about going and doing loads of exam practice, because the kind of answers that you will get on exam papers will come up again and again. Make a list of the kind of ethics that they have suggested on past exam papers for each of those particular areas of the question. Have yourself a list of ethics suggested for IVF, ethics suggested for stem cell research, ethics for Daphnia, ethics for this, ethics for that. Because a lot of people dislike it so much that they actually don't revise it, they actually don't learn it, and I would recommend that you don't fall into that trap. Right, there are some quite interesting questions in this bit. If you get a cystic fibrosis question, people tend to expect to get a question about how cystic fibrosis actually affects the person or how the mutation itself actually works. They don't necessarily expect to get a question like, suggest why cells from mouth swabs or blood samples are used rather than gametes. And it can throw you a bit. But it is a little bit of common sense, actually. You've got to let yourself just keep an open mind and use some common sense. Obviously, it's easier. It's less difficult for men to provide a gamete sample, but for women, if you're going to be testing her gametes, it's going to be an incredibly painful process. Not only that, gametes are only haploid. They will only contain half of the adult's genetic information. So if you're testing half of the DNA, you're not getting the full picture. If you're testing for several different recessive alleles, well, maybe you might think, actually, maybe there's more than one allele that causes cystic fibrosis. Maybe it results from many possible mutations in many possible DNA, chromosomes and so on. In the risk analysis shown, if neither partner is a carrier, it's considered the chance of having a child with cystic fibrosis is low, not 0. This throws people crazily. It throws people because people know that if neither are carriers, then it will be 0. We believe that these monohybrid inheritance situations are absolute, and that if you are both homozygous dominant, then you will only have homozygous dominant offspring. Okay, that's not necessarily the case. The reason being that these mutations occurred at some point in the past, there's no reason to say that they can't occur again. They might occur. That's why it's low and not zero. Scientists are extremely cautious. We never, ever, ever confirm a theory, for example. We can reject a theory if we have evidence against it. But even if we've got exceptional quantities of evidence for a theory, we never, ever confirm it. There could be something wrong with the test. A low proportion of tests have mistakes in them. False negatives and false positives are a big deal when you're talking about testing for genetic disorders. So make sure you know what both of them are and make sure that you're able to use them in the right contexts. If the partner is found to be a carrier, then they do offer screening to other family members. And why is this screening offered? It's basically if one family member is affected or a carrier, then there's a potential that other family members may be affected as well, and that they should have the same opportunity to make informed choices as the original couple do. There are ethical problems with that, and that's going back again to ethics. Make sure that you know all of the ethics about all sorts of things, and I'm going to add genetic screening to that list as well. Now, E is a monohybrid cross. Now, there is a very, very standard breakdown of marks for monohybrid inheritance crosses. So make sure you have got all the aspects of all of these things into your answers, because there is a really, really predictable way that they allocate marks for this. The first one is you have to state the genotypes of both the parents. So I would have parental genotype, and it has got both of the parents are heterozygous. So you've got, it doesn't say which particular letters to choose, so just choose whichever one you want. I would choose a capital C and a small c. Okay, so there's your first mark there, the parental genotypes. The second one is then, it sounds really ridiculous, but it is, they actually give you a mark for separating those genotypes into the gametes. Now, I've got quite an old-fashioned way of doing it. You can either do it like that if you're going to do the sort of drawn crosses, or if you're going to do it as a Punnett square, as titles for your Punnett square. So either method of writing the gametes is acceptable for that second marking point. Next bit is to get the clearly writing out the genotypes of the offspring. So in the Punnett square, that is just filling out this table there. Now, notice down here, some people worry that if they write the capital C and the small c in the wrong order, then they will be penalised. Don't worry, you won't be. It's fine. You can write a small c followed by a capital C. The other way that I always learnt to draw my genetic crosses is this. I'm just drawing it very, very quickly. I'm not going to explain it in great detail, but I'm just going to draw it here to show that if you are one of those people who's learnt to do it this way, then that is acceptable as well. Okay, right, so again, the genotypes need to be known. Now, the fourth mark again is for the phenotypes of these offspring. So you can do this in various different ways. You could either write the phenotypes in the Punnett square or underneath the genotypes themselves, or you could write the number of offspring with that particular phenotype. So you could say 1 unaffected, 2 carriers, and 1 sufferer. People will generally, if you use slightly different words, it should be fine as long as it's clear which one is which. Then you finally, because it has specifically asked calculate the probability of having a child with cystic fibrosis, you do have to actually calculate the probability, which then is quite simple. It's either 1 in 4, 25%, or 0.25. Right, now we're on to the last question here, question 8, about obesity and the BMI. Now, the first one is very, very simple. All you've been asked to do is calculate BMI. You've even been given the equation, so the only thing I would suggest here to do is to actually show you're working out. So with 2, the next bit is using the information in the table, interpret this man's BMI. It's a 2-mark question, so you cannot just say he's overweight. You have to say he's in the range 25 to 29.9, therefore he's overweight. Or you could say he is overweight, but only just. So notice that it's a 2-mark question and act accordingly. Another classic describe question. compare the effect on BMI on relative mortality for men and women. For both of them, it decreases and then increases. So that is a comparison, because you're saying it does the same for both. There isn't much difference until you get to a BMI of 25, and then after that the risk for men is greater than the risk for women. So you're not really, there isn't much to say in terms of differences, but remember, even when you're comparing, you don't just talk about differences, you talk about similarities as well. Right, so in C then, the next question, the relative mortality of 1.2 or less indicates a low risk of dying. And you can then look at your BMI for the woman of 32.5, follow your graph up 32.5, that line of best fit basically, and follow it along there, and you can then tell what her relative mortality is. So it is 1.25. So she doesn't really need to be concerned about her risk of dying with her obesity. It's just a case of can you read this graph in an effective way. The final question then is about cardiovascular disease, and it's talking about, it is again a quality of written communication question, but this particular one is describe 2 changes that this woman may be able to make to her lifestyle to reduce her risk. Explain how each change would reduce the risk. So this is really a very useful paper for me to be able to show you the different kind of QWC questions, because this is the other type of common QWC question where you've got to link concepts here. If we were going to go with the idea that she could eat less food, then you could say the woman could eat less food or go on a diet, because that would help to reduce her BMI and therefore hopefully reduce her chance of getting CVD. Reducing cholesterol levels, because cholesterol is associated with atherosclerosis. Reduce your saturated fat, because that reduces your blood cholesterol. All of these are pairs of points. So with this one, you've got to have pairs of points. If she smokes, she should reduce it, because smoking increases the risk of atheroma. If her diet should reduce the salt levels, because high salt is associated with high blood pressure. Lower her alcohol intake, because high alcohol is associated with high blood pressure. So hopefully that will help you. If you've got any other questions about anything else, then please let me know, or your tutor or whatever. So good luck with your work and enjoy.